Hey guys, welcome to your first edition of Virtual Notes. So that we don't get a day behind, since we already did test corrections yesterday, I want to make sure that we cover what we need to cover today. So I'm going to go through the PowerPoint and take notes pretty much how I would in class. On each slide, make sure you pause the video so that you can write down the information you need to have from each slide, and then listen and add in extra information to the side of your 2-column notes. So it's going to be pretty much the same, except I'm there, but I'm not technically there. So for today, we're going to talk about cell transport. And we've talked about cells and what the different organelles within cells are. But if we have a cell here, and it has its nucleus, its nucleolus, you have all the different organelles, all of this inside. The key part we're going to focus on in this unit is the outside cell membrane, because we know from our definitions of organelles that the cell membrane is what controls things going in and out of the cell. So when we talk about cell transport, we're talking about how can we get things in and out of the cell through different mechanisms. So we start out first as a reminder of what the cell membrane is. Now this is not on your 2-column notes, this is from the slides from last time with cells, but I want to pull it back up just so we remember what it's like. The cell membrane is a very thin, flexible barrier, which means it moves. It's going to regulate what enters and leaves the cell. It's going to provide protection for the cell and a little bit of support, but the main support in a plant cell would be the cell wall. It is selectively permeable. Now, that term selectively permeable is really important because it comes back and it means it lets some things in and some things out. So, you guys are teenagers. You might hear your mom or dad say that you have selective hearing, meaning you hear some things, but you don't hear everything. You kind of pick and choose what you hear. It's the same thing with the cell membrane, that it will pick and choose what it lets in and what it lets out within the cell. Now, when you look at the parts, you have the lipid bilayer. So if I draw this cell, we have phospholipids, and the phospholipids have heads, which are what I'm drawing right now. You might want to draw this on your page. And they have tails. And you have 2 tails coming off of each head. Hold on, I'm getting there. Okay, so we have the lipid bilayer. So we have our phospholipid heads and our fatty acid tails. So these are the tails. These are the heads. And we have essentially 2 areas. You have the heads up top and then you have the tails in the middle. Now, sometimes you're going to have these proteins that go across the entire membrane. And they're going to play a role in allowing things in and out. So this is a protein. We also have these little carbohydrates on the outside of the cell membrane. I was drawing these little dots coming off here. So this would be a carbohydrate, and that carbohydrate is going to be like an ID tag, telling other cells what kind of cell the current cell is. It's going to allow them to identify one versus another. So that's your basic structure of the cell membrane. You have the phospholipid bilayer, you have the heads, and then you have the tails. There's proteins within it, and there's carbohydrates on the outside. Now, if we want to look at a better picture than my little drawing, this would be a better picture than my drawing, but you still get the point. What we're going to have is you have the heads up top up here. You have the carbohydrate ID molecules, which are in the yellow. Then you have these proteins that go across the whole cell membrane. So this would be the inside of the cell, and this would be the outside of the cell. Extracellular means outside of the cell, and then the cytoplasm is on the inside of the cell. So things are going to pass back and forth through this cell membrane, and that's what this whole unit is going to focus on. Now, the fluid mosaic part of this model is something you might want to write to the side of yours. Fluid means that everything moves, and then mosaic means that there are a lot of parts. Of parts. Okay, so with the fluid mosaic, we have your carbohydrates, we have proteins, we have the phospholipid heads and tails, And so all of these parts work together to make the cell membrane. But think back to the inside of the cell video I showed you, how it was all moving. And all of these little pieces are fluid. They move from different locations. And that's what helps the cell membrane be really productive. Okay, so there's different types of movement through the cell membrane. The first one we want to talk about is called diffusion. Diffusion is where particles move from a high concentration to a low concentration. Now, the way to remember your concentrations, I want you guys to picture a slide. Okay, so if this is the slide, somebody normally sits up here on the slide and they go down the slide. Okay, now diffusion is where particles move from a high concentration to a low concentration. So the example would be this person up here going from high at the very top of the slide to low at the very bottom of the slide. The key thing here is it doesn't require energy. You sit on top right on that cusp of that slide and you're going to slide right down, no problem. That's diffusion, moving from high to low. Now when we're talking about high to low, it's not necessarily location of going from up top to bottom, but it's meaning from high where there is a lot to low where there is a little. So if you look at this diagram over here, you notice on this side we have a lot of green. So this would be considered a high concentration of green, and over here would be considered a low concentration of green. Now, if this barrier, this cell membrane in the middle, the orange, if this is permeable to green, meaning green can get through, it's going to go from high to low. They are going to spread out. We don't like molecules when they're really close together and there's a ton of them. If we can spread them out, we will. Now, the movement going from high to low, where we move from this side to this side, that is diffusion. Now, we're going to move until we hit this key term, which is called equilibrium. This key term, equilibrium, is going to mean that it is equal amounts on both sides. Okay, equal amounts on both sides. So diffusion is going from a high concentration to low. It's going to go until you hit equilibrium, where it's the same amount on both sides of the membrane, and it does not require energy. Things just move from high to low. Kind of like you're at top of the slide, no energy to go down to the bottom of the slide, and you went from high to low. That's your key for diffusion. Think about if you have a perfume or somebody in the hallway sprays Axe, because that stuff smells. If you're out in the hallway and somebody sprays a little bit and you're cleared out at the other end of the hallway, those particles, when they first spray it, are really, really highly concentrated. There's a ton of them. But then they're going to spread out very, very slowly until it is in equilibrium. Now, eventually it's going to reach you at the end of the hallway because no one can escape the Axe musk. But when it gets down to you, it's not as strong as it is of where they first sprayed it. That's because it's reached it either has reached equilibrium or it is very close to where it is spread out really far as it can go. That's diffusion. Our next key term is osmosis. Osmosis is diffusion of water, and that's going to be your key term right there. Same concept of diffusion, but this time we're talking about the movement of water. So with water is going to go through a selectively permeable membrane. Remember, selectively permeable means that some things get through, but not all things get through. So when water goes through diffusion, again, it requires no energy because it's going from high to low. Remember the slide, high to low, no energy. Where we have a lot of water on one side and it's going to go to the side where there's not as much water. And it's going to do this, again, until it hits equilibrium. Now, there are three major solutions when we talk about osmosis. Sorry if you heard that noise. That was my dog. He likes to scratch. Okay, so three different types of solutions. We have isotonic, hypertonic, and hypotonic are our three major solutions when we deal with the movement of water. Isotonic is where solutions on both sides of the membrane are equal, and so nothing changes. It's the same thing. So a way to remember this, I'm going to give you some stupid ways to remember this, but it works. Okay, isotonic, don't want to change. So don't want to change is an isotonic solution because there's no net movement anywhere. It just stays the same. Hypertonic is where we have a higher solute concentration outside of the cell. And when we have a higher solute concentration, that means less water outside of the cell. And so since there's less on the outside of the cell, the water on the inside of the cell is highly concentrated versus the outside of the cell, so water is going to leave. So cells are going to shrink is going to be your key thing there. Now, hypertonic, I think of the word hyper here. And hyper, the way I remember this, is if you're really, really hyper, you have this burst of energy, and then you shrivel up and die. What I mean by shrivel up and die is you don't necessarily die, but you lose all your energy, you pretty much collapse. Kind of like a sugar crash, where you get the burst and then you're done. Okay, so that's what I think of hypertonic. Now, hypo is the opposite. Hypo is where there's a higher solute concentration inside the cell, which means that there is less water on the inside of the cell. Okay, it's key to note where the water is. On hypertonic, there's less water outside of the cell. On hypotonic, there's less water inside of the cell. So the water on the outside is high concentration. The water on the inside is less concentration, so it's going to go from high to low, and the cell is going to swell. Okay, hippo, it's going to make cells fat like a hippo. Okay, now let's put some pictures to this so you can see this. Okay, with this one, we have two different scenarios here. You have your animal cell and you have your plant cell. The reason that we did this is because they react differently to the different solutions. We're going to start off in the middle. Now, by the way, this picture is on the last page of your notes. Okay, isotonic here is normal. This is the solution you want to be in, where there is no change in the size of the cell. Now, that doesn't mean there isn't movement. That's a key thing that you want to include here is water still moves. It's not just going to sit there. It's still going to move back and forth. But when it moves, it's an equal amount of movement. So you'll have water coming in and water going out. Water in, water out. Now, with your isotonic, remember, iso don't want to change. So the cell size stays exactly the same as normal. Okay, that's generally easy to remember. Iso don't want to change. Now, if you have a hypertonic solution, so we're moving to the right here. Hypertonic, remember hyper is where you get really excited and then you shrivel up and die. Okay, so you'll notice here my animal cell is all shriveled and my plant cell has shriveled. But it looks different here. Now, before we get into the difference, let's talk about water movement. What has happened is all the water has left the cell. So there was higher concentration of water inside of the cell, less on the outside, and osmosis says water moves from high to low. So it moved from high in the cell to low outside of the cell. From high to low. Excuse me. And we end up with a shriveled little cell. Hypertonic, they shrivel up and die. Now, they react differently. The boundary on the animal cell is only a cell membrane, so it's going to shrivel. But on a plant cell, we have this cell wall, and then we have the cell membrane. What you're going to see in a plant cell is the cell membrane will pull away from the cell wall, and that's called being plasmolized. Okay? The cell wall doesn't change, but the cell membrane and the cell itself is going to shrink. Then you have your hypotonic. Hypotonic is where water is going to rush in, and it's going to burst it. It's going to make it fat like a hippo. Now again, same thing. Animal cells don't have a cell wall, so their cell membrane will end up breaking and the cell will become lysed. What we mean by lysed is they burst, and they break and they die. Whereas a hypotonic for a plant cell is going to push against those cell walls. Notice how they're rounded. It's going to push against those cell walls, and it's going to expand the cell. But that cell wall is really rigid, and it's not going to actually burst. So you have isotonic, iso don't want to change. Hypertonic, we're going to get really hyper, and then we're going to shrivel up and die. And then hypotonic are going to make cells fat like a hippo. Okay, the next type of diffusion, so far we had regular diffusion, we had osmosis, and now we have facilitated diffusion. Now, you're facilitated. A facilitator is someone who allows something else to happen. So in this scenario, we're going to use protein channels that are going to move molecules across because they can't just go straight through the cell membrane. We still go from high concentration to low concentration, just going down the slide, high to low, but it does not require energy, just like diffusion and osmosis. The only difference is that we have to go through a channel to be able to get through that cell membrane. That's it. We need some help. We need a helper. Then we have active transport. Active transport, the word active, if you're active, that means you're doing something. Well, in order to do something, you are going to require energy. So active transport requires energy. Here's the opposite. It's going to go from low to high. That's the key thing right here. So if you think back to our slide example, this is our slide. Okay, that's our little ladder. This person's standing down here, and they want to get up the slide. Okay, in order to do that, they would have to run up the slide. That's going from low to high concentration. So you're going from low to high, and it is going to require energy. If you have a slide that steep, it's going to take a lot of energy to get all the way up that thing. So active transport is going from low to high, and it requires energy. So if you were to look at this taking place, you would have, down here you have a low concentration, so not very many, and a high concentration up here, and we're moving them up from low to high. Since you're going against the concentration gradient, and that's a key statement to write down, against the concentration gradient, we're going from low to high, so we're going against the flow, we are going to have to require energy. Because so far in diffusion, facilitated diffusion osmosis, we always go from high to low. So we're going against the flow here. So that is our active transport. It requires energy, and that energy is in the form of ATP. That's the energy that we care about. Okay, a form of active transport is called endocytosis. Endo means inside, so it is inside of this, taking things into the cell. Now, it's taking large things into the cell, because small things can just go straight through the membrane, no problem. It's the large things that have issues. What happens is the membrane is actually going to surround it, and it's going to create its own vacuole, and I'll show you a picture of that, of how that happens. Now, this is used for large molecules, mainly food or liquids. So there's two different types. You have phagocytosis, which is taking in food, and pinocytosis, which is taking in liquids. Those are your two major types that you need to know. Here's a picture of endocytosis. You have things that are outside of the cell that needs to get into the cell. The cell membrane is actually going to come in, and it's going to essentially pinch off and form a vesicle on the inside. And by doing that, you're taking these materials that were once on the outside here, they're going to come in, they're going to get pinched off, and now they are on the inside of the cell. So it's things that can't cross through, and we need to get them into the cell. Now the opposite of that is exocytosis. Exocytosis is taking large amounts of materials outside of the cell. So it's where you have your vesicle here. It's going to come over to the cell membrane, and they are going to fuse together, and it's going to push out its materials. So endocytosis is getting things into the cell, large things. Exocytosis is getting things out of the cell. Okay, that's your basics of your types of movement within the cell. Remember the key things to remember are your different terms, and then does it require energy or not? So is it active transport or is it passive transport? Okay guys, I'll see you tomorrow. Have a good night.